Alright, so lab 10, 3 different exercises, and we actually have you refer to one of the other ones for some pictures. And I have demo plates of almost everything today that we'll also take a look at and compare to your unknown organisms. So we have Mannitol Salt Agar. Those are the pinkish kind of plates that we see and have the abbreviation MSA on them. We have the Columbia CNA with 5% sheep blood. So those are the blood plates. So those are the red plates, look like blood because they do have whole red blood cells in them. And then the eosin methylene blue with lactose agar is the purplish plates that have the abbreviation EMB for eosin methylene blue on them. Both of these media, all 3 of these media, have 2 properties to them. They're both selective and differential. Selective media favors the growth of one type of organism while inhibiting the growth of another. So typically it has something in it that's toxic to a particular group of organisms and stops their growth, right? So we'll identify the selective agent in each of these 3 media that we're looking at today. They're also differential, which helps us distinguish, just like gram stain and acid-fast stain are differential stains, right? Endospore and capsule help us see those special structures. This is going to tell us something about the organisms, and mostly it's going to tell us biochemical characteristics, things that your organism can do. And based on what it can do, it's going to create either color changes in the growth of the organism or in the media. And so we'll look at changes. We'll even look at your organism, and we'll also look at the media itself for changes. So the first one we're going to talk about is Mannitol Salt Agar, abbreviated MSA. And this is selective for what group of organisms? Anyone know? Mostly Staphylococci in clinical specimens. And this is because they are salt tolerant. The selective agent in this media is salt. But it's not just the presence of salt, because all media for the most part is going to have salt. It's the fact that it's a high concentration of salt. So it's 7.5%. This is 8-fold higher than normal saline, right? That's isotonic solution of 0.9%. Right, so this is a very high salt concentration. So for your organisms, because we're not just looking at clinical specimens, right, stuff that you would get from a person, some of the organisms you guys are working with would come from soil and things like that wouldn't commonly necessarily contaminate somebody's wound or something like that. So if your organism grew on this plate, you know that it's salt tolerant, right? That's what we know about it. And we can assume it may be a Staphylococci. But again, we have other organisms in this lab, so that does not ensure that you have a Staphylococci if you have growth. So most of you guys will notice, though, no growth on these plates, right? And when you look for your organism, only a few of you guys will actually have growth. All the rest were killed by that high salt concentration. This media is also differential. Anybody know what the differential agent, what they have to use to create the different? Phenol red is a pH indicator, but what component are they actually eating? The mannitol, right? It's a sugar alcohol. And some organisms can ferment this sugar. Now we know this already, guys. What are some common byproducts of sugar fermentation? Acid, gas, one more. Our favorite on the weekends, alcohol, right? Sometimes during the week, depends on the week. So, which one can we detect in this media? If they're fermenting, right, as we said, they're going to produce these things like acids, alcohols, and gases as they break down this sugar and use it for energy. in a process we commonly know as fermentation. Our pH indicator. And we've dealt with this one before, right? This is in our phenol red tubes. So we already know the color changes that could happen, right? So like the phenol broth test, phenol red is the pH indicator in this media. Don't have to learn a new set of colors. These are adjusted to a little bit different pH, which is why they're kind of pinkish. And that over time they get kind of pink as opposed to the orange tubes we kind of started with the phenol red. So what color indicates acid byproducts? Yellow. Yellow. Right? Just like in our phenol red test, yellow means low pH, means acid. So this is a really nice streak plate done on the Mannitol Salt Agar. And you can see where there's a lot of organisms, they produced a lot of acid, and it all turned yellow. But notice here where there's a little bit more organisms, it's not quite yellow yet, it's that in-between color of orange. You would, of course, look where you have the most growth and the fact that your media is yellow. Only one organism should do this for sure in our class, and only one of you guys had it. Right? So one of you guys has one of these plates that looks like this, all yellow. What's the name of the commonly found clinical specimen that's going to have these attributes? It's going to be a Staphylococcus, it can grow in the presence of this high salt, and it can ferment mannitol. What species? Staphylococcus aureus. So who's got Staph aureus? Who's got the yellow plate? That's you. So Carolyn's got Staph aureus. I bet before today you knew you had a staph, but you weren't quite sure which one, right?
Well, you told me last time.
Oh, I did. Okay. You were probably able to narrow it down with your methyl red VP because they came out right. Cool. So we didn't do this because you guys have already seen your organisms growing on Nutrient Agar, right? In your slants or TSA might have been the regular growth media. Why would you do that? Why would you do just a regular growth media and grow your organism? What purpose would that serve in an experiment? Just to see the normal growth on normal growth media. What do you guys think? A control, right? As a control. We didn't do that because, again, you've already seen your organism growing under those conditions, right? Instead of using more plates. If you remember, right, from the beginning, most of your organisms, right, were kind of clear looking or creamy. There was only a few of you guys that had pigmented colored organisms, right? So the colors that we'll see later, you know, are from this special media, are not your organism. Normal look to it. And also this would help us confirm that your organism could grow, right, that it is in fact the salt that killed it. What would be the likely consequence of omitting the salt? What if we didn't have any salt at all in this media? This media would no longer be what? Yeah, this salt, this high concentration of salt makes it selective. So it would no longer be selective. And therefore, just about any organism would grow on the media. So you could have some false positive results, right? If the media wasn't made properly. Now, taking that into consideration, what is that for this? Is this a specificity or sensitivity issue? How many people think specificity? How many think sensitivity? And the rest are unsure, too afraid to raise their hands. It's a specificity issue, right? Because that salt helps us be specific as to what we're selecting for. That high salt And it's going to help us hopefully only grow the organisms that have that high tolerance. And in clinical specimens, that's mostly going to be Staphylococci. These are the organisms that grow on your skin, right? Sometimes our skin is a very salty environment, so it makes sense. We talked about this before when we talked about carbon and nitrogen sources for which one? Citrate or phenol red? Which one was really evil? The citrate, right? There was only one carbon source, and what was that? It was a citrate derivative, sodium citrate. Okay, and there was only one nitrogen source in that media. For this media, if you look at the different food sources, beef extract is one of the ingredients. This is animal cells, right? So they can contain all elements that you could need for life. So it definitely contains both carbon and nitrogen. Peptone. These are protein derivatives. They contain both carbon and nitrogen. So plenty of stuff for these guys to eat. And there's even a special sugar in the alcohol form, mannitol. Sugars, on the other hand, are just carbon and water, right? Carbohydrates. So no nitrogen, just carbon and H2O. So they got plenty of carbon and nitrogen. That's not an issue. That's not why they're not growing, right? They should grow. The problem is that high salt concentration. Now, could you do this single test? If someone gave you a specimen, could you put it on mannitol salt and know right away that it's Staphylococcus aureus because it turns yellow and it grows? No, right? It could be something else. But in a clinical specimen, chances are you're probably looking at Staphylococcus aureus. This is I can't ever say this word. Presumptive test, right? You would presume, but you would do further testing. For you guys, right, we did a whole bunch of tests leading up to this one test. And this is more of a confirmatory for you guys. Where in a lab, they might start in the opposite direction. They might put it on this plate and then run the additional tests. Because this would help narrow it down for them, right? Make sense? Okay. So, what do you see on this plate? You see growth? Yeah, that's actually growth. Right? Of course, I asked them to do a streak plate and they couldn't follow directions. You guys seen this picture of this plate before on a quiz. They just put a new inoculum of bacteria in each quadrant instead of dragging from one quadrant to the other. But that's okay. We've got growth, right? So, what can you tell me about this organism? The fact that it can grow on this media. What type of ability does it have? It's salt tolerant, right? It can go in the presence of salt. Okay. Do we see any color in the changes in media? What color are we looking for with this media? We're looking for yellow. We definitely don't see yellow, right? It's pink. So it probably didn't ferment, right? The mannitol. So we know it's salt tolerant, right? And it's a non-fermenter of mannitol. Everyone agree with me on that? It could be a Staphylococci, right? And in fact, I know what was put on this plate. This was Staphylococcus epidermis, right? So this one, unlike aureus, does not ferment the mannitol, right? But it will grow. It is a staph. But all you would be able to tell, unless you had other testing, is what we just said, right? The growth indicates it can grow in high salt concentrations. No yellow, right, indicates that it cannot ferment the mannitol. So it could be Staphylococcus epidermis, right, or some other staph other than Staph aureus, if you're dealing with a clinical specimen. Even then, they do further testing. And so we've already answered this, right? What would you see if it was Staph aureus? You'd see yellow. You'd see growth, and you'd see that yellow. So this plate has Staph aureus on one side and Staph epi on the other, which is what I tried to do in your guys' control plates that you have on your desktop, except one of the plates I screwed up and put Staph aureus on both sides. But also because these plates are kind of old, we get this bleed over where you're getting yellow on the other side. But you should hopefully would have seen, right, yellow on just one side, just for the Staph aureus. And growth on both sides, because both are tolerant to staph. So this plate, this is the best picture I could get. Do you see how the plate itself is still kind of pink? Maybe a little orangey over here, but it's not, the plate itself is not yellow, right? The media itself is not yellow, but the organism has turned yellow, and it's growing. So the fact that it's growing, it's what? Salt tolerant. Just because the organism turned yellow does not mean that it's fermenting mannitol. The media actually has to turn yellow, right? Because that's where the pH indicator is. Why some of these organisms turn yellow when they grow on this plate, I think is maybe an adaptation to growing in the high salt concentration environment. So, does anybody have a plate like that? That they have growth on the mannitol salt for their organism? I know it might be Jade's plate that she's out. Is it 108? Okay. Can you? Yeah, that one's getting a little yellow around it, but that one is not. That's not a staph, right? And again, I think it's due to the age of the plate, and maybe over time that one might ferment. I don't know. That's not an organism that we would deal with in a clinical lab. It's a bacillus. It's a one from soil. Right? It's a soil contaminant. So why are we performing this experiment? Right? We want to know if your organism is salt tolerant. Halophilic is a term we use actually for salt requiring organisms, or salt tolerant, or halodoric sometimes referred to. You'll see that terminology. And again, some of our bacilli can be salt tolerant, and we wouldn't normally see those in a clinical lab, though. Among the survivors, if it grew, then we can ask the next question: can it utilize mannitol, right? And that we detect by the color change in the media. So this is pretty easy, right? We spot inoculate a plate, we stick it in the incubator. We look at it 1 to 2 days later. These I refrigerated for us, so hopefully they wouldn't overgrow. We can get any false positive results. The media surrounding the colonies remains red, means no fermentation, right? Media surrounding the colonies turns yellow, means that you have really good fermentation of that sugar and the acid production. So growth organisms are salt tolerant. It's adapted to a high salt environment. If the media turns yellow, the organisms utilize the mannitol to generate acid, right, which is a fermentation product. And so Staphylococcus aureus will do this. And also some of our bacillus, right, will have a little bit of this fermentation possibility sometimes. So the next plates, these are my favorite ones. Actually, I like them all, but this is one of the cooler ones. So these are ones we actually buy from a manufacturer. We don't pour these ourselves because they have whole red blood cells from sheep. Sheep blood acts similar to human blood. I guess to not drain the blood bank, we're doing this testing. Sheep work just as well. This also contains 2 antibiotics, and that's what the CNA stands for. Columbia is a type of media, base media that they use for this test. And what this test allows us to do is detect the presence of what are called hemolysis. And these are exoenzymes that some bacteria produce, and they can cause different degrees of lysis to the red blood cells. So actually cause direct damage to the membrane of the red blood cells. So why do some organisms produce this? Think about this. A red blood cell, what's inside that red blood cell? Food, right? Nutrients, proteins, there is a particular element that's in a really high concentration in our red blood cells that bacteria are also after because they need it for their metabolism. So elements are things like carbon and nitrogen, oxygen. What one is in really high concentration in your red blood cells? Iron. It's the iron they're after. Red blood cells are a very rich source of iron. So this media is selective for what group of organisms? Gram-positive or gram-negative? Gram-positive. So only if you had a gram-positive, I gave you one of these plates, right? The rest of you guys have gram-negative organisms. I didn't even bother giving you one of these plates. Because what does this plate contain that's going to stop the growth of gram-negatives? 2 antibiotics. Colistin and nalidixic acid. And that's why it's the C for colistin and Na for nalidixic acid. These guys inhibit the growth of gram-negatives. So if you had a gram-negative, it wouldn't have grown on these plates. So like if you had E. coli, it wasn't going to grow. If you had a gram-positive, you got one of these plates and you should have hopefully got growth. So notice colistin and E. coli both begin in coli. That's kind of a clue there, right? That gram-negatives like E. coli are killed by colistin. A little play on words. Whether it's like that on purpose, I don't know. This is awful differential. So what differences are we going to see? What is in this media that's going to be changed by the organisms? What are we looking for? We're looking for hemolysis, right? And what are they going to do to red blood cells? They're going to lyse them, right? So unless you have whole red blood cells, this media isn't going to work, right? So you need red blood cells, whole red blood cells, not broken. This is the differential agent. This is what's going to be changed by the bacteria. That's why these plates look blood red. They literally have blood in them. These are the 2 types of hemolysis that you're going to see. And these are best observed when you hold the plate up to the light, right? And so beta, which how I remember this one, I always remember beta is bad because this is the worst case scenario. It completely lyses the red blood cells. When you hold up these plates, you can see right through the plate. They've completely annihilated the red blood cells around their growth. The other type is what we call alpha, and this is incomplete lysis. This is where they poke holes in the red blood cells, and oxygen gets in, and you get this green discolorization. So if you hold up the demo plate that I have, right, there's all different types on there. There are ones that didn't affect the red blood cells at all. There's 2 different types of beta. One where the organism really lyses. You can see that big clear zone. And then another one where you can see the organism is growing and you can see around it the big clear halo of where it killed the cells. And then there's one that's kind of greenish. So I'll come, like I said, hold it up to the light. And so I'll come around to each table when you guys are looking at your plates, and we'll talk about those demo plates. So what happens is that the iron combines with the oxygen, you get that greenness discolorization for the alpha. So here's some pictures of ones I've done in the past. So again, notice that greenish tinge, right? And this one I had fun with it, right? I even drew an alpha. So there should be one spot on that demo plate that looks like that. Over time, it'll start getting brown. Now that's when I did the best I could to take a picture of it. So again, right, do you see the clearing around the growth? This is complete lysis. So this would be what? Alpha or beta? This one's beta, right? Is this organism gram-positive or gram-negative? It's got to be positive. Why? Because there's growth, and gram-negative is what? Get killed by the colistin and nelodicid, the two antibiotics, right? So Staph aureus again, this is a special one. If you can kind of see the backwards handwriting there, right? This was a Staph aureus plate. So, Carolyn, does your blood agar plate look like that? Do you have beta hemolysis? Yeah. Hold it up to the light, see? Oh yeah. And then some organisms, right, are what we call gamma hemolysis. I hate this terminology because it sounds like it's lysing the red blood cells, right? Gamma actually means that it does not lyse the red blood cells, right? This does not cause damage. And so you can see on these two plates, you can see the bacteria growing, but notice the red blood cells are unchanged, right? They're still red. You don't see green. You don't see halos. So no hemolysis, no damage to the red blood cells. And that's true for a few of you guys' organisms as well. It'll grow, but won't cause any change to the red blood cells. So this plate is one of my favorite ones. It's a nice streak plate of a gram-positive organism, except we've got a problem. What is that right there? That's hemolysis, right? Which one? Alpha or beta? Beta. It's completely clear, right? Is that the person's organism, you think? No, what is that? That's contamination. And probably literally from breathing on it. Staphylococcus aureus, a lot of people have in their noses. And so breathing on the plate could have deposited a few of those organisms and caused them to grow. And then on this plate, you can see the beta hemolysis. So yeah, if the whole plate was like that, then that would have been her organism, right? But that's clearly a contaminant. An interesting contaminant at that, though, right? Good teaching moment. So why would we do nutrient agar plates? Why would we do that? What would they act as? A control, right? And again, your organisms should grow on those plates, no problem. And you guys have been growing on Nutrient Agar and TSA, your growth should be fine. That media doesn't inhibit or change the growth at all. So early formulations of Columbia CNA agar were made using a slightly higher concentration of colistin and nelodicic acid. So these are the two antibiotics, right? But this was reduced to improve recovery of gram-positives. What's some possible reasons why a high concentration was less effective at allowing the gram-positives to grow? So the problem here, right, lots of antibiotic. And so it was even killing off some of the gram-positives. Why would that happen? Why would a high concentration be a problem? When we did our Kirby-Bauer test, right, we talked about what is that concentration that we want. We want a minimum inhibitory concentration, right? We want to know the least amount of the antibiotic we can use that will still kill the organism. And in this case, we definitely don't want it killing our gram-positives. So concentration really matters, and especially with colistin, it affects membrane integrity. So remember, the gram-negatives have got that outer membrane and the inner membrane, so they're very susceptible to antibiotics that deal with membranes. So again, in too high of a concentration, even our gram-positives could be negatively affected by that antibiotic. And nelodicic acid interferes with DNA replication. And all cells do DNA replication. If this can get into the gram-positives, it could cause a disruption for them as well. So these aren't strictly antibiotics necessarily for gram-positives versus gram-negatives in how they work if you don't use them at the right concentrations. Right? So concentration really matters. Because remember, anything in too high of a concentration will kill you, like water, right? If you keep drinking and you don't pee, it will kill you. So this is really important. Emphasis again on the concentration of antibiotics, right? And we can go into really specifics on them. We're not going to. So, colistin, remember, membranes. And as I mentioned a moment ago, gram-negatives have got that double membrane, right? So they're more susceptible to this antibiotic. It's hydrophilic. It loves membranes, so it's really attracted to these membranes and helps distort them. So like alcohol, remember that affects membranes, so does colistin. So gram-negatives are very susceptible to this. Too high, though, you're going to damage your gram-positives too. So what is this test going to do? It's going to help us isolate gram-positives. So again, I only really gave it to you guys that have gram-positives. And typically we're looking at staphylococci and streptococci and enterococci. And then, does it affect the red blood cells? Right? So we just inoculate plates. We come back, we look at them for their results. Beta is complete lysis, right? Clearing through our plate. Alpha is just partial lysis. And then gamma, remember, is no lysis. What does this tell us about our organisms? We have growth. It should confirm you have a gram-positive, right? Clear halos. It completely lysed the red blood cells. That's what we call beta. Greening means that it partially lysed, right? It's what we call alpha. No destruction of the red blood cells, right, would be gamma. All right, our last one. And I'm kind of bummed because my demo plate doesn't have my favorite organism showing up well on it. But one of you guys have it, and it's still nice and green, so we'll pass around your plate. Actually, I think it's your plate, Rain. No, not Raina. Who has E. coli? Who's got E. coli? Nobody that's here? The plate's out there. Somebody's got it. We'll find out in a moment. So typically this type of media is used for a general category of organisms referred to as coliforms, right? These are a broad class of bacteria. It's not a classification. They are found in warm-blooded mammals in their feces, so in your intestines, your large intestines. A large number of them are lactose fermenters. So they're gut microbes, they're gram-negative, they're bacilli, right? And they can ferment lactose and produce acids. And so that's what we're looking for today as ones that would fall into this category. So, is this media selective? Yeah. For who? Gram-negative, right? So if you had a gram-positive, it should not have grown on this plate. We had plenty of this plate, though, so I gave it to you anyways. What in this media stops the growth of the gram-positives? It's the eosin and methylene blue. Those two dyes are toxic to the gram-positives. So who's not going to grow? Gram-positive. So if you had a gram-positive organism, hopefully your growth was inhibited on these plates by those dyes. So coliforms are gram-negative, right? So we want gram-negatives to grow. So what component is the differential agent? What are they going to change? What are they going to eat? What are they going to ferment? The sugar. What's the sugar in these plates? Lactose or sucrose? The formula that we use in lab only contains lactose. There's different formulas you can buy of eosin methylene blue, right? The one we use just contains lactose. So we'll only know if it's a lactose fermenter. Anybody remember what sugar was in our phenol red tubes? Glucose. Glucose. So E. coli is a fecal coliform. What color would you expect the growth to be on this media? This is the pretty one. You'll get a black or metallic green growth. And this is the interaction of the acid it produces when it ferments and the dyes that are present in the media. So somebody's got to have a plate. Look at your EMV plates. Who's got the pretty green plate? It's out there. I know it is. I saw one. Now, you guys, back table? Not yours, is it? No, I know what yours was. Maybe it's still here. Maybe I'm hallucinating. I'll find it and show you guys. It's around somewhere. My control plate, unfortunately, it didn't stay. So if the growth of an organism ferments lactose but produces a small amount of acid, what will the growth look like? So less acid, less interaction with the dye, so we're going to get some different colors. What are the other colors we might see? Purple and pink. So see this one? This was actually from some hamburger meat one semester. Give you an idea of the kind of coliforms that could be present in that if you don't cook it all the way. And see, that's showing the metallic green. Here's one that's showing kind of a purplish color. And this one, it's kind of blending in with the plate, but you'll see growth. And that's because it's a non-fermenter. So what's the purpose of the Nutrient Agar? Control, right? So we could compare. We wouldn't necessarily do that in a clinical specimen. So the dipotassium phosphate is a buffer, right? And remember, we're dealing with acid-base situations. We want to adjust it to a particular pH when we start. What would be the consequences if we had the pH at starting a little too high, at 7.8? That means that they have to produce more acid, right, to get color changes. So this would potentially affect our results, right? We would need a lot more acid produced to get that darker color so we could get some inaccurate results. So pH is important, especially when you're doing a pH-related test. If someone messed up the pH of the media, would this affect the media's sensitivity or specificity? The pH. Sensitivity. Again, it's going to make it hard to tell the color changes and be accurate if the pH isn't in the right range. So, if it's metallic green, is it definitely E. coli? No, right? It's a pretty strong indicator that it may be E. coli, right? If you're an Untitled Chapter and it looks like a penguin, it probably is a penguin, right? So in most cases it'd probably be E. coli, but it could be something else. So again, we would always do further testing. For you guys, right, the one that is metallic green really is E. coli. So these organisms are growing on both of these plates. And they're kind of blending in with the plate, so it's kind of hard to see. So we can tell one thing about these organisms because they're growing, right? Are they gram-positive or gram-negative? They're gram-negative. because gram-positives won't grow. What kills them? The dyes, the eosin and the methylene blue. Right? So, are they fermenting? No, because what colors should we see if there's fermentation happening? Pink, purple, black, or metallic green. None of those you see, right? So it's not a fermenter. These, I know it's kind of hard to see, but they're growing, so we know they're gram negative, right? What color? They're kind of a pinkish purple, right? So, a lot of fermentation or little? Little, right? A little bit of fermentation. So, gram negatives, remember, are going to grow. Gram positives are not. Then we can ask, if it grows, is it fermenting the lactose? And the degree of fermentation is determined by the different colors that you see. Right? So we put bacteria on the plate. Right? Colorless, no fermentation. Pink, modest level of fermentation. Purple or black or metallic green are really strong fermenters, right? Usually indicative of an organism like E. coli. So for our organisms that you should hopefully see fermentation for is definitely E. coli, that metallic green and black. Enterobacter, if you had that one, it should grow pinkish purple. Klebsiella pneumoniae, this one's kind of cool. It looks really dry when it grows on this media. I don't know why that is. It looks very different texture-wise than it does on Nutrient Agar. So it's kind of interesting one. So again, the color is going to indicate the level of fermentation. No color change to the, and we're actually looking at the organisms for this one, not the media, because they take up those dyes. Yeah, I'm going to come around